Yeah, Mark, you can introduce Andres.
Will do. Before I do that, I think we have a few people on Zoom who are not muted, and my earbuds are going crazy. So please make sure that you mute yourself if you're attending remotely. And with that, I would like to take this opportunity to introduce Andres Mayorga Gomez. Andres is a PhD student in my lab, but he initially came to the University of Georgia from Columbia to work on his master's degree with Dr. Nambisan. And after finishing up his master's degree, Andres joined my lab, where he is working at the intersection of lighting and water use efficiency. And with that, Andres, I'll turn it over to you.
Thank you. So today I'm going to be talking about crop growth and water use efficiency in controlled environment agriculture. Controlled environment agriculture, and part of the operation expenses in controlled environment agriculture are really related to the electricity use. For instance, in greenhouse we have a supplemental light, and the annual electricity cost could account like for around 30% of the average annual farm gate value in vegetable greenhouses. And in some specific cases, in greenhouses, you have to pay, greenhouses pay around like $200,000 per hectare per year just in electricity costs. In the case of indoor farming and vertical farming, we have, it is estimated that around the 50% of the operation expenses are related to the lighting systems and the humidification systems. And roughly there is like a 25% for lighting and 25% for the humidification systems. So you can see here that the amount of money that this kind of these setups have to pay just in electricity is really high. So we are exploring like the possibilities and how to reduce, if we can, like how to reduce the electricity expenses in controlled environment agriculture. So first, I'm going to start talking about supplemental lighting greenhouses. So it has been for multiple and for different crops. It is the amount of light that crops need every day has been calculated to grow like in an optimal way, and so this is why we normally use, or people normally use supplemental lighting greenhouses, because not always it is possible to achieve this amount of light, or the target, or the DLI, the daily light integral for each crop, just by having like the light from the sun. For instance, when there is like a shady day, we are not getting like the same amount of light in the crop, or if we are in a specific season of the year. For instance, in winter we have different, in winter we have short days, and in the summer we have long days. So in order to achieve this amount of light that the plants need to grow properly, and if the sun is not, if we cannot get that from the sun, we use the supplemental light. So in a recent study made by Thesis and Mark from our lab, that was called Reducing Energy Requirements for Greenhouses Lighting Through Carryover of Excess Light to the Next Day, they found that plants can tolerate small DLI changes, fluctuations, after the plants are, when the plants have like a day with high DLI. They found that the next day you can provide, like, the plants can tolerate like a reduction of on the DLI that they are receiving. So they also found that when these DLI fluctuations are big, the plant growth is affected. So also as I said before, they found that by following, when you provide like a little bit, when you provide like less light, like when you reduce the DLI after having a day with high DLI, plants are not like negatively affected. And by using like this strategy, for instance, like you have one day with high DLI given by the sun, and the next day you have a low DLI, and you think that you have to provide like the supplemental light. This is not always like necessary. You don't always have to do this, because the plant is going to the plant growth is not going to be like affected if like that fluctuation is small. So with this approach, it has been they have been calculated that you can save like around $6,000 and $7,000 per acre per year in depending on the location of the crop and the weather conditions. So with that said, we wanted to determine the effect of fluctuating DLI levels on lettuce growth, and more specifically, we wanted to see if a crop receives more than the required DLI on one day, can growers provide less light during the following day or days without a negative impact on growth. So in the previous experiment, they just measure like the DLI fluctuations between the 2 days, but in this one, we were trying to see if we can reduce the DLI for the crop like during multiple days. So in order to do this, to test this, we grew two lettuce cultivars in a walking growth chamber under controlled conditions, and we have a different. Our treatments were a combination between a combination of different. Our treatments were a combination of one day with high DLI, like that exceeds the target or like the reported DLI for lettuce, and followed by a different number of days with low DLI levels. But always we keep like the average DLI of 15 mol per square meter per day, which is like the one that is recommended for lettuce growth. So in the control treatment, we have like all of the plants receiving like DLI of 15 each day. And just to, I mean, like the treatments are a little bit complicated, but this is like one of the treatments that we had like one day with high DLI, like exceeding like the 15, that is the target DLI, followed by a day with low DLI, and the average DLI between those 2 days was 15. The next treatment was one day with high DLI, followed by 2 days with low DLI. The average DLI for those 3 days was 15, and the rest of the treatments were the same, but having 3 days, 4 days, or 5 days with low DLI following the day with high DLI. So always having the same average DLI of 15. So one of the results that we got from this study is that we found that when we have just 1 day of low DLI, the final dry weight of the plants was reduced, or like the biomass accumulation was reduced. From this experiment, we conclude that lettuce growth is reduced by 13% with just 1 day with low DLI, and the lettuce growth is not significantly reduced when plants are under multiple days with low DLI. So this information is important to take into account when planning like the strategies for supplemental light in greenhouses, because according to this, at least in lettuce, we can like play and we show that we don't have like to always apply or like provide like the target DLI when we reduce like the DLI for multiple days without affecting the plant growth. Now I'm going to talk about the humidification and the indoor cultivations. And as I said before, like in vertical farming, the 50% of the operation expenses are related to the light, and the 50% and the light and the humidification. And so like around 25% of those expenses are only for the humidification process. So why do we have to use like the humidification process in these setups? Is because vertical farming is like a closed system, and all of the water that plants transpire gets into the air, and this humidity or this water vapor has to be removed by the air conditioning system. If we don't do this, the humidity of the growing place is going to increase, and this increase, like for instance, the susceptibility to diseases. With that said, we wanted to first to determine the effect of different DLI levels on plant water use efficiency, and how is water use efficiency defined. First, in the leaf level, the water use efficiency is the ratio between the photosynthesis and the transpiration of that leaf. And in a plant level, the water use efficiency is defined as the biomass that the plant can produce by unit of water. So in this case, the dry biomass in grams per liter of water. So we grew up one lettuce cultivar again in a walking chamber under controlled conditions, and in this time we changed it, like the light intensity. We had like different treatments. We have different treatments of light intensity from 125 to 375 micromole per square meter per second, and we use the portable photosynthesis machine in order to take measurements of the water use efficiency and the photosynthesis in the leaves of these plants. As a result, we got that the net photosynthesis increased when we increased like the light intensity, and the transpiration increased, but the transpiration didn't increase as fast or as much as the net photosynthesis did. And as I said before, the water use efficiency in the leaf level is given by the ratio between the net photosynthesis and the transpiration rate. So the water use efficiency also was increased, or was higher when we increased the PPFD levels for the plants. So here we have like kind of a trade-off, because when we increase like the light intensity of the lights, we produce the plants grow faster, and the water use efficiency is higher too. But when we provide like a higher light intensity, so we have to use like more energy, but since the plants are growing like faster, so like the growing cycle is short. So if the growing cycle is short, so we have like the plants during like a less amount of days under those conditions, and there is like a less amount of water that we have to remove from the air, and possibly, I mean, maybe reducing at least like the expense of the energy that we use for removing the water from the air. So what are we going to do next? So like the previous study was just, was just in the leaf level, and so a leaf might not be just like a representation of the whole plant. So we are just going to see like what is like the responses of the leaf and the whole plant. So in order to do this, we are going to take the same measurements, like the photosynthesis and the water use efficiency in the whole plant, and the photosynthesis we are going to take it by using the gas exchange chamber, and the water use efficiency we are going to estimate the transpiration and the water use efficiency by using load cells, by measuring like the water loss of the plant, like in multiple, during a period of time. And we are going to see how these values, or how this is related to the leaf case, which is like we are going to take again the same measurements that I was talking about before, like with the CIRAS-3, the photosynthesis and water use efficiency, to see how they relate. And finally, we want to see like what are like the crop responses. So we are planning like to take the same measurements during the whole growing cycle. So first, we are going to again use the plant water. We are going to calculate the plant water use by using load cells. Oh, this is going to be again in the working chamber, like that's trying to imitate like the vertical farming conditions. We are going to calculate the plant water use with load cells, and through imaging, as you can see in the right, we are going to estimate the canopy size during different parts of the growing cycle, at the same as the plant water use, in order to see how, like, what is the relationship between like the canopy production and the use of water. And we are going to do the same with the total dry weight to see what is the relationship between the biomass produced and the water used. And finally, like what we want to is try to estimate the energy used for lighting and the dehumidification process to see. I mean, I was talking about a trade-off effect, that we were like providing more light, and we have like to use more energy providing more light, but we are going to reduce the energy for the dehumidification process. So we want to estimate the energy used for both to see. I mean, to see if we can, like, if we are reducing the energy consumption in general in these kind of setups. So, any questions?
Could you go back to slide 16?
Yeah.
Okay, so it looks like there's not a linear relationship between transpiration and your PPFD, just based on your R squared. Do you have any idea why that might be?
I mean, for me, looks. I mean, for me, is that like the transpiration is like really similar, no matter what is like the PPFD that we are applying to the like the light intensity that we have, right? So I mean, that's why the water use efficiency is being higher when we have like high PPFDs, because the transpiration is like, I mean, I'm not saying that it's not increasing, but increases like really slowly, but the photosynthesis increase a lot.
I have a question. I'm sorry if I missed this, but have you developed a relationship, or is there an established relationship between transpiration and relative humidity? So, I mean, if you're looking for an ideal range of relative humidity for disease control, but like if you optimize disease control versus reducing transpiration based off of relative humidity?
So can you repeat that? I cannot hear you well.
Is there a relationship between relative humidity and transpiration?
Yeah.
And how would that play into, you know, indoor growing based on what you're trying to control for disease?
Yeah, I think that there is a relationship between the humidity and the transpiration. And what I was talking about the diseases, I mean, we have to reduce that humidity, like, for sure, you know. So like, I mean, we are not like trying to control. I mean, I'm not trying to control like this. I'm not trying to do like a control of diseases, but like the thing is that we always have to reduce that humidity. So I mean, I don't know if I understood your question like correctly, but I'm not. I mean, I'm not like trying to do like a control, like a disease control with this stuff. I'm just like trying to see if I can just like reduce the amount of energy that we need to reduce humidity, that we are going to have to do that in this kind of environments. So like if we have like the plants, like the growing cycle, I don't know, let's say like 40 days for lettuce. And if we reduce it like to, let's say 3-5 days or something like that. So we always have like to reduce, we always have to take that humidity out, no matter like what. So
But it's always 50, I mean, 60%, and then you have.
oh, what I was talking about, the 50%, when I was talking about the humidity, I was talking about like the 50% of the operation costs in this kind of environments
Oh, okay.
is like just for the electricity that you need for lighting and for the humidification. That's like the 50%. So like it's calculated like maybe roughly 25% is for like 25% of the expenses in these kind of crops is for just electricity in lighting, and the other 25% is the electricity, but for the humidification.
If anyone online, if anyone has questions.
Hey, Andre.
Yeah.
Can you hear me?
Yeah.
This is Leo Lombardini. I have a question. This, we're done at 800 ppm CO2, right?
Yeah, can you repeat that?
Yeah, sorry, my microphone is not working very well. This data that you're showing now on the screen, we're done at, we're collected at 800 ppm CO2. Is that correct?
Yes.
Do you think that if you had run it at regular CO2 concentration, your graph would have been different? And if you think so, how so? How different?
I mean, it could have been like if it was different. Photosynthesis.
Yeah, if you run the same, collect the same, measure the same data at 390, 400 ppm. Do you think that transpiration, especially transpiration, would have been the same, or you think that it would have been different?
He was like, I believe his answer was like, hell yeah, get after it. Yeah, and he was like, just.
Okay. If we reduce the concentration of CO2, I mean, this is I'm gonna try to make a educated guess here. I would say like the transpiration would be similar, but the net photosynthesis could be reduced because when you reduce the ratio, when you reduce the CO2 in the environment, so the plant, so you are going to have like more, maybe you could have like more oxygenation processes instead of like just carboxylation processes in order to make like CO2 fixation to the photosynthesis. But I mean, I think that the point of having this, I don't know like what would be the point of doing this with a lower concentration of CO2 if in vertical farming, that's one of the things that we can do, right? Like just provide more CO2 to improve the carboxylation rates and increase the photosynthesis.
I'm thinking about the effect of CO2 concentration on stomatal conductance and stomatal aperture. So that's why I thought it'd be interesting to see the difference. But
yeah, I mean, honestly, I don't know what the stomatal conductance here. If we reduce the CO2.
okay, thanks.
Any other questions from online?
Do you guys have any other question?
Yeah, any more questions from online?